Okay, so today we are going to start our new chapter from microbiology, and this is a very important chapter for your GPAT and other exams. So this sterilization chapter is again important chapter, and lot of questions they used to ask on this chapter. If you have seen previous papers, you may find few questions every year from this chapter, sterilization. Now this chapter is common in your the chapter is common in your microbiology, as well as this chapter is also present in the pharmacology. The chapter is also given in the pharmaceutics. So we are not going to discuss separately. So sterilization, what do you mean by the process sterilization? Sterilization मतलब क्या होता है कि sterilization means absence of any viable form of microorganism or removal of any viable form of microorganism or killing of any viable form of microorganism. The process of removal or killing of all viable forms of microorganism, including their vegetative form and spores. तो माइक्रो ऑर्गेनिज्म का डिस्ट्रक्शन जो है उसको अपन बोलेंगे स्टरलाइजेशन. डिस्ट्रक्शन कौन सा? सभी टाइप की माइक्रो ऑर्गेनिज्म. वाइबल फॉर्म मतलब क्या है? वाइबल मतलब जिंदा. वाइबल मीन्स जिंदा. वाइबल मीन्स लिविंग. सो all viable forms of microorganism, vegetative form and their spores. Spores are not a viable form, but they are a hibernated form. So they can regrowth when there is favorable condition. So destruction of or killing of or removal of all viable form of microorganism, including their vegetative form and spores, known as sterilization. Now, why sterilization is there? Sterilization is very important. For example, if you want to do some surgical operations, and if that utensil, if that scalpel, if that forceps, they are contaminated with some bacteria, or for example, say for example, your tetanus bacteria, so they may cause tetany to the patient. Suppose whatever injections you are using, if they are contaminated, so they may cause disease to you. So that's why sterilization is very important to ensure the accept to ensure the absence of organisms, because they causes the disease. So sterilization is nothing but the process of removal or killing of all viable form of organism, including their vegetative form and spores. Now aseptic technique is nothing but the technique by which now it is followed in the pharmaceutical way. It is followed in the pharmaceutical industries, those which are engaged in the production of parenterals. So this technique is the process in which all probable contamination of microorganism that can be prevented to get the sterile product right from the beginning. एक दो तरीके होता है. एक तो जो है आप माइक्रो ऑर्गेनिज्म है आपके अगर उस प्रिपरेशन में, तो उसको आपको डिस्ट्रॉय करना है बाय यूजिंग स्टरलाइजेशन. दूसरा एक प्रोसेस होता है कि आपको वो माइक्रो ऑर्गेनिज्म का कंटेमिनेशन आने ही नहीं देना है आपके प्रोडक्ट में. तो आने ही ना देने के लिए आप कौन सा टेक्निक फॉलो करते हो? aseptic technique. So right from the beginning, right from the raw material, right from the process, right from the place, so you are avoiding the contamination so that final product will be sterile, known as the aseptic technique. So it is a technique for production of large volume parenterals. It is a technique or process in which all possible contamination of microorganism can be prevented to get entered into your product being processed. That is known as aseptic technique, right? So that is followed in the industry for large volume parenterals or simply injectables. Now, what do you mean by disinfectant? What do you mean by disinfection? As the name indicates, disinfection. So whatever microorganisms are there, that is being killed by using chemicals known as a disinfectant. Disinfectant again. So there is a difference in the sterilization and disinfection and sterilization. The destruction or removal of all pathogenic organisms capable of giving rise to the infection. Chemical disinfectant, they are safely applied to a skin. Chemical disinfectant which are safely applied to our skin or mucous membrane and used to prevent the growth of microorganism is known as a antiseptic. So chemicals which can kill the microorganism, they are known as a disinfectant. Disinfectant जो होता है, disinfectant, it can kill the microorganism. Disinfectant are the chemical which kills the microorganism simply. Now, what do you mean by antiseptic? Antiseptic is again chemical. Antiseptic is again chemical which prevent the growth of microorganism. It will not kill. It will prevent the growth. That's why there is a difference. Antiseptic and disinfectant. Normally, disinfectant is applied on inanimate and inanimate objects. डिसइंफेक्टेंट किसके लिए अप्लाई करते हैं? जैसे फ्लोर है या फिर कोई आपका यूटेंसिल है ना, जैसे आपका बीकर है, डिसइंफेक्टेंट. जैसे आपको कोई एपरेटस है, डिसइंफेक्टेंट. बट डिसइंफेक्टेंट आप स्किन के ऊपर नहीं अप्लाई करते. स्किन के ऊपर आप अप्लाई करते हो एंटीसेप्टिक. So disinfectant is a chemical which can destroy the microorganism, which can kill the microorganism. Antiseptic is the chemical which prevent the growth of microorganism. It is not killing the microorganism. I hope I am audible to you. Now, different methods of sterilization. Different method of sterilization. So there are different methods which are used, which are utilized for the killing of microorganism in the industry, in the pharmaceutical industry, as well as in the laboratory scale. So method of sterilization. So it may be either physical method, just by using sunlight or drying. So you might have seen grains. आपके घर में क्या करते हैं दादाजी दादी? तो वो जब समर आता है तो क्या करते हैं, सब जो घर में का जो अनाज है वो सब आप कहाँ पे लेके जाते हो? वो गच्ची पे लेके जाते हो, वहां पे सब स्प्रेड करते हो, है ना, धूप खाने के लिए. क्या होता है उसमें? सो योर सनलाइट हैज अ यूवी रेज. Your sunlight is also having UV rays, and that UV rays they are killing the microorganism present in your grains. So that is a physical method. आप जो है ना धूप में रखते हो आपका अनाज, क्यों? For the sterilization. So physical method, right? Now again dry heat. Dry heat. So dry heat means destruction of microorganism by using dry heat. So you are using flaming or incineration or hot air. इंसनरेशन देखो आप, इंसनरेशन मतलब जब आदमी मरता है, मरने के बाद में क्या आता है कि उसकी बॉडी जो है ना, वो बॉडी वो सड़ने लगती है. when a person will die, his body will decompose, right? And during the decomposition, lot of organism, microorganism, they will grow on your body, so they may spread the infection to the nearby people. That's why कि आप जब मरते हो तो आपको जलाया जाता है. You are subjected to incineration. Why? Because of that flaming. You might have used this flaming for your microbiology practicals. Suppose if you want to isolate something, so you might have isolated microorganism by using loop. So before isolation, before inoculation, you are sterilizing that loop by just passing through the flame. So flaming. Hot air. Next, that is a moist heat sterilization. Okay, another method is moist heat sterilization. Moist heat मतलब क्या? यहाँ पे water vapors रहेगा. Water vapors at a high temperature. So water vapors, okay, water vapors has a more penetration power than dry heat. Okay, so moist is, so that is involved in the pasteurization. Pasteurization is the process of sterilization of milk. जैसे आप milk के लिए pasteurization करते हो. You are heating up milk at a 80 degrees Celsius, boiling, steam under normal pressure, steam under reducer. Next one is nothing but the filtration. So bacteria-proof filters are used. So those filters which are having pore size less than that of bacteria. जो बैक्टीरिया प्रूफ फिल्टर्स होते हैं उसमें जब फिल्टर करेंगे आप तो बैक्टीरिया ऊपर ही रहेगा, फिल्ट्रेट में रहेगा. So filter candles, okay, then asbestos pad, then membrane filters. Membrane filters are they are made from a cellulose. इसके ऊपर भी क्वेश्चन आ चुका है. Membrane filters they are made from cellulose. Membrane filters are made from a cellulose. And for sterilization by filtration, membrane filters are used. Radiations, okay, so UV radiations, then non-ionizing and ionizing radiations, ultrasonic radiations, sonic vibrations, they are also used for the killing of microorganisms. UV, IR, ultraviolet rays, non-ionizing, beta rays, alpha rays, gamma rays. Yeah. So they are also there. These radiations they also kill the microorganisms. So methods for sterilization, these are the methods for sterilization. Dry heat method है ना, moist heat method है, filtration है, radiations है. Second, by using chemicals. Chemical method. Chemical method में क्या है? Alcohols. They are used as a sterilizing agent. So ethyl alcohol, isopropyl alcohol, then trichlorobutanol, aldehyde like formaldehyde and glutaraldehyde. Aldehyde भी है, जैसे ऑपरेशन थिएटर में क्या होता है? ऑपरेशन थिएटर में बिफोर एनी सर्जिकल ऑपरेशंस, सो दैट थिएटर मस्ट बी स्टरलाइज्ड, फॉर दैट दे आर यूजिंग दिस फॉर्मालिटी। तो जो हॉस्पिटल में है ना, आजकल हॉस्पिटल में देखो, जब ऑपरेशन करना है तो अगर वो उस टाइम पे देखो, आप जो ऑपरेशन होने वाला है बिफोर दैट, सो दे यूज्ड टू स्टरलाइज द होल ऑपरेशन थिएटर, है ना। उसके उस वहाँ पे जब आप एक बहुत ही बैड स्मेल आता है because of that sterilization. What they are doing? They are using this formaldehyde or formalin solution. So formalin or formaldehyde, and in that they are mixing potassium permanganate. तो formaldehyde plus potassium permanganate, उसमें क्या है कि फॉर्मेलिन का फ्यूम्स निकलता है, that fumes sterilize whole operation theatre, formaldehyde, glutaraldehyde. Then again there are other chemicals also, like surfactants like phenol, dyes, okay? And gases like ethylene oxide, okay, formaldehyde, then beta propanolactone. These are the examples of chemical methods, okay? तो देखो फिजिकल मेथड होता है पहला, उसमें है ना, उसमें ड्राइड मॉइस्चर, है ना, बाद में फिल्ट्रेशन, रेडिएशंस, और बाद में नेक्स्ट मेथड है आपका केमिकल मेथड, इसमें अल्कोहल्स, एल्डिहाइड, बीटा, बहुत सारे डिफरेंट डिफरेंट chemicals use करते हैं. Next is nothing but the sterility. What do you mean by sterility? Sterility is nothing but the total absence of any form of microorganism from your preparation. कि आपका प्रिपरेशन में कोई भी कंटेमिनेशन नहीं है, आपके प्रिपरेशन में कोई भी वाइबल फॉर्म नहीं है, वेजिटेटिव फॉर्म नहीं है, स्पोर्स नहीं है. Absence of any form of microorganism from your preparation known as sterility. आप बोलते हो स्टेराइल है, स्टेराइल मतलब क्या? Nothing is there in that, okay? So nothing microbial grow microorganism or their any kind of any form is not there. So not, okay, it is nothing but the total absence of any form of microorganism. Antiseptic क्या होता है? Antiseptic is nothing but the chemical, antiseptic is nothing but the chemical which is capable of preventing the growth of microorganism, and it is applied it on the body, it is on the animate. So it is applied on the animate, मतलब वो अपने बॉडी के ऊपर अप्लाई कर सकते हैं. केमिकल which are capable of killing microorganism or preventing the growth of microorganism when, and it is applied on the body, human body. Disinfectant is the chemical which kills the microorganism, but it is not applied on a inanimate object. It is applied on a inanimate object, not on a human body. तो एंटीसेप्टिक ह्यूमन बॉडी के ऊपर अप्लाई किया जाता है टू प्रिवेंट द ग्रोथ ऑफ माइक्रोऑर्गेनिज्म और टू किल द माइक्रोऑर्गेनिज्म. तो डिसइंफेक्टेंट जो है दैट इज अप्लाइड टू डिस्ट्रॉय द ऑर्गेनिजम्स, माइक्रोऑर्गेनिजम्स on the utensils like floor, tables, etc. As I told you. Next we'll see how you can different terms which are used to describe the sterility, or how you can validate that your product is sterile, or how you can know whether microorganisms are decreased or not. So these are the different terms which are used to indicate the destruction of microorganism. So बहुत ही इंपॉर्टेंट टर्म है, इसके ऊपर क्वेश्चन आ चुके हैं, 2 बार क्वेश्चन आ चुका है. द डेसिमल रिडक्शन टाइम, the decimal reduction time, or it is also known as d value. Decimal reduction time, decimal reduction time or d value, or also known as it is nothing but the decimal reduction time or d value of the surfactant is nothing but the time of disinfectant required to reduce the population of microorganism by 90%. It is nothing but the time for a surfactant which is required to reduce the population of microorganism by 90%. मान लीजिए आपके इसमें कुछ 100 बैक्टीरिया है, तो अगर एक आप में इसमें एक डिसइंफेक्टेंट है, उसके लिए कितना टाइम लगता है टू डिक्रीज द माइक्रोऑर्गेनिज्म बाय 90%. दैट इज नोन एज द थर्मल डेसिमल, ओके, डेसिमल रिडक्शन टाइम, दैट इज द डी वैल्यू. क्वेश्चन आ चुका है इसके ऊपर. 1 डेसिमल और 1 डेसिमल d value of disinfectant is nothing but it is the time of surfactant required to reduce the population of microorganisms by 90%, or 1 decimal. 1 decimal, that is 90% of 1 logarithmic unit. That is known as d value. Next होता है thermal destruction time, z value. Thermal destruction time or z value. Z value of disinfectant, that is the degree of temperature required to reduce the 1 log of d value. इसके ऊपर क्वेश्चन आ चुका है. What is Z value? The degree of temperature required to reduce the 1 log of d value. मतलब क्या है? Simply degree of temperature required to change thermal reduction time 10 fold. 10 गुना आपको thermal reduction time change होगा. So thermal destruction time is nothing but the Z value. Thermal decimal, okay. The decimal reduction time is nothing but the D value. Decimal reduction time is nothing but the time required by disinfectant to decrease the 90% of microorganism. Thermal destruction time is nothing but the, it is a degree of temperature at which there is a 10 fold changes in the D value. और इसके ऊपर क्वेश्चन आ चुका है 10 fold changes in the D value. That is to reduce the 1 log of D value. Different method of sterilization. Different method of sterilization. बहुत सारे methods होते हैं. So out of that, dry heat, moist heat, radiation, and disinfectant. They are most commonly used. So dry heat sterilization. Dry heat sterilization. By using dry heat sterilization, आप क्या sterilize करते हो? You are sterilizing your utensil. You are sterilizing your pipette, burette, etc. You are sterilizing your broth also, right? So dry heat sterilization, you are using hot air oven for that. You are using hot air oven. So I have not shown this diagram of hot air oven here because you might know, you might have seen hot air oven. Dry heat sterilization is done in a hot air oven. Remember. Now, what is important? Holding temperature. That is important. So what is the holding temperature in the dry heat sterilization? Dry heat sterilization में holding temperature कितना होता है? याद रखना है holding temperature कितना होता है? 180 degree Celsius for 2 hours. एक होता है 180 degree Celsius for 2 hours और 260 degree Celsius for 45 minutes. तो dry heat sterilization में hot air oven में holding temperature कितना होता है भाई? 180 degree Celsius for 2 hours. 180 के ऊपर कौन सा माइक्रोऑर्गेनिज्म जिंदा रहेगा और 260 डिग्री सेल्सियस फॉर 45 मिनट्स. तो हॉट एयर ओवन में है ना. सो मैकेनिज्म होता है. व्हाट इज द मैकेनिज्म फॉर किलिंग ऑफ द बैक्टीरिया? बैक्टीरिया मरता कैसे इतने टेम्परेचर? मतलब क्या बैक्टीरिया को क्या नुकसान पहुंचता है, कैसे मरेगा? सो यूजिंग ड्राई हीट so that dry heat causes the oxidative damage and denaturation of bacterial protein. Bacteria is made from a cell. सेल के अंदर क्या होगा? उसके एंजाइम्स होंगे, उसके उसका है ना, उसको प्रोटीन है. सो ऑक्सीडेटिव डैमेज टू द बैक्टीरियल एंजाइम्स, डीनेचुरेशन ऑफ बैक्टीरियल प्रोटीन. मैकेनिज्म याद रखना है, क्वेश्चन आता है. व्हाट इज द मैकेनिज्म ऑफ ड्राई हीट स्टेरिलाइजेशन? क्वेश्चन आ चुका है. ऑक्सीडेटिव डैमेज एंड डीनेचुरेशन ऑफ बैक्टीरियल प्रोटीन. सो एप्लीकेशन क्या है? सो ड्राई हीट इज यूज्ड टू स्टरलाइज द ग्लासवेयर्स or other metal instrument, powders, oil injections. They are sterilized by dry heat. Next to the moist heat sterilization. Moist heat मतलब क्या है? यहाँ पे water vapors. Okay? Moist मतलब water. So moist heat sterilization. As the steam has a more penetration power than dry heat. So moist heat sterilization is used. So apparatus is not about the autoclave. Autoclave आपने देखा होगा लैब में वहाँ पे. Autoclave is used for the moist heat sterilization. Now what is the holding temperature? 115 to 117. 115 से लेके 117 डिग्री सेल्सियस फॉर 30 मिनट और 121 डिग्री सेल्सियस फॉर 15 मिनट. So 121 degree Celsius for 20 minutes. 121 degree Celsius for 20 minutes at a 15 psi. So that is the holding temperature for moist sterilization. At this, steam with the high pressure, okay, which causes the denaturation and coagulation of bacterial protein. याद रखना है, this is the mechanism of moist sterilization. Coagulation and denaturation of bacterial protein. तो important है holding temperature and mechanism. Right? Moist heat sterilization is used for the IV fluids, rubber, plastic container. Okay. Then next to that, tintilization. Tintilization is also known as a fractional sterilization. Tintilization is also known as a fractional sterilization. Question आ चुका है इसके ऊपर. So what is the holding temperature? Heating of a medium at 80 degrees Celsius for 1 hour, 100 degrees Celsius for 20 minutes for successive 3 days. 3 दिन तक आपको हीट करना है 80 डिग्री सेल्सियस के ऊपर 1 आवर के लिए या फिर 100 के ऊपर 20 मिनट के लिए. डेली 3 days you have to heat the medium for 3 days. So that tintilization, it is nothing but it is used for the sterilization of your ventilation. Fractional sterilization is known as a fractional sterilization. So it is used for the surgical dressing and culture media. So important है ये चार्ट वाइज किया है मेरे साथ यहाँ पे चार्ट वाइज है इम्पोर्टेंट है इसमें मतलब यहाँ पे तीन ही रोज है तीन कॉलम है लेकिन कम से कम 4 या 5 क्वेश्चन यहाँ तक अभी तक इसके ऊपर आ चुके मतलब यही इस तरह और नेक्स्ट वन इज द पाश्चराइजेशन व्हाट इज द टेम्परेचर ऑन पाश्चराइजेशन इसके ऊपर क्वेश्चन आ चुका है सो टेम्परेचर फॉर पाश्चराइजेशन व्हाट डू मीन बाय पाश्चराइजेशन इट इज अ मेथड ऑफ स्टरलाइजेशन ऑफ योर मिल्क मिल्क आप जो स्टराइल करते हो मिल्क सो मिल्क इज made sterile by using pasteurization. What is the pasteurization? What is the holding temperature? Holding temperature is nothing but the 62.8 degrees Celsius. कितना है 62.8 degrees Celsius. That is 63 degrees Celsius for 30 minutes. It is known as a holder method. अगर temperature है आपका 62.8 degree for 30 minutes, it is known as a holder method. Second भी होता है इसमें holding temperature 71 to 72 degrees for 15 seconds. 71 to 72 degrees for 15 seconds known as a flash method. Flash method. Third temperature होता है इसमें 140 डिग्री लेस for less than 1 second. So pasteurization के लिए तीन टेम्परेचर होता है 62 डिग्री सेल्सियस 62.8 degrees for 30 minutes then 71 to 72 degrees Celsius for 15 seconds. That is a flash method and more than 140 degrees less than 1 second. That is known as a pasteurization. It is used for sterilization of milk, popularly known as a pasteurization of milk. Next होता है radiation sterilization. Radiation sterilization में क्या है? Different radiations use करते हैं, light radiations. So first one is the UV radiation. So UV ultraviolet rays are used. So it can be produced by mercury vapor lamp, okay, or hydrogen discharge lamp or any other method. Right? So radiation in the range of 253 nanometer they can damage your nucleoprotein of the microorganism. UV radiations क्या करते हैं they will damage a nucleoprotein they will damage the DNA they will damage the nucleoprotein of the bacteria mechanism याद रखो they will damage the nucleoprotein or DNA of the bacteria एक होता है UV ultraviolet okay it is used for thermolabile substances maintaining the aseptic technique also used in the hospitals. So UV chambers are available for this. Second होता है आपका ionizing radiation. So ionizing radiation मतलब alpha rays, beta rays, gamma rays. So they denaturate the and damage the DNA. तो alpha rays, beta rays क्या करते हैं? They damages your DNA, an enzyme, and other component of microorganism. Formation of free radicals by forming the free radicals. So gamma rays and beta rays are used as a ionizing radiations. They are used for thermolabile substances, syringes, plastics, catheters, needles, surgical dressings and blades. They are sterilized by ionizing radiation. Third होता है आपका gaseous sterilization. Gaseous sterilization. So in that different gases are used. First है आपका ethylene oxide, then formaldehyde, then beta propanol electron. These gases are used. So all they are the alkylating agent. मतलब क्या ये मिथाइल इथाइल रेडिकल बनाएंगे और बाइंड करेंगे न्यूक्लियोप्रोटीन को because the alkylation of hydroxyl sulfidyl group of bacterial protein they will kill the bacteria so they are used for thermolabile substances plastic rubber fumigation operation room sterilization and fumigation of aseptic room. So these are the different methods for sterilization okay? So different methods for sterilization in short sterilization by filtration is known as a cold sterilization याद रखना है sterilization by filtration is known as a cold sterilization question आ चुका है इसका now for that you are using bacteria proof filters यहाँ पे क्या है bacteria proof filters आप यूज करते हो है ना पहला है आपका filter candle filters और filter candle so they are made from unglazed ceramic filter or diatomaceous earth filter then asbestos filter इसके ऊपर एक कमेंट है they are carcinogenic in nature then sintered glass filter prepared by fusing the finely powdered glass particle. They are prepared by fusing finely powdered glass particles. Next आता है membrane filter. Membrane filter आता है membrane filters they are made from cellulose. मेम्ब्रेन फिल्टर सेलुलोज से बनता है. Am I audible to you? Yes or no? Am I audible to you? So next one is a membrane filter. So sinter glass filter, they are made by fusing the powdered glass particles. Right? Sintered glass membrane filters made from cellulose and other polymer. Now radiation sterilization में non ionizing radiation low energy type UV/IR होता है. They will damage to a genetic material. Ionizing radiation so high energy रहता है इसमें. X-ray रहता है, gamma rays, cosmic rays. So they have a little effect on genetic material and other vital component of microorganism. Sterilization by filtration. Now chemical sterilization by using different chemicals. By using different chemicals or disinfectant. पहला है alcohol. What is the mechanism of alcohol? So ethanol is used. Isopropyl alcohol is used. So IPA is most frequently used in the industry. Isopropyl alcohol सबसे ज़्यादा इंडस्ट्री में यूज किया जाता है. इथेनॉल है ना, आपने देखा आयन alcohol based sanitizers. So they are most frequently used. They act by denaturating the bacterial protein and they have no action on spores. अल्कोहल क्या करता है, बैक्टीरियल प्रोटीन को डीनेचुरेट करता है ना. अगर आपका प्रोटीन डीनेचुरेशन हो गया तो दैट इज यूजलेस, सो बैक्टीरिया विल डाई. जैसे बैक्टीरिया का प्रोटीन डीनेचुरेट हो गया सो इट इज यूजलेस टू बैक्टीरिया, बैक्टीरिया विल डाई. नेक्स्ट वन इज aldehydes. So formaldehyde is there, so it will interact with the amino acids of the protein, amino acid of the protein of the microorganism, and mostly used as a preservative for anatomical structure. तो आपने अगर है ना, आप अगर कोई एमबीबीएस कॉलेज में जाओगे तो वहाँ पे जो बॉडीज है, बॉडीज एमबीबीएस कॉलेज के जो 12th में भी देखा होगा आपने, you might have seen in the 12th also. So different bodies organ, they are preserved in the glass bottles by using formaldehyde, formalin solution. So it is used to preserve the anatomical specimens. Formaldehyde gas is widely used to disinfection of operation theatre. Phenols होते हैं, so lethal effect, their killing effect is because of their capacity to cause membrane damage and releasing cell containing to cell. So बैक्टीरिया है, बैक्टीरिया के अंदर क्या है, सेल. सेल मतलब सेल के अंदर न्यूक्लियस है, साइटोप्लाज्म है और सेल वाले. सो फिनॉल क्या करते हैं, दे कॉजेज द डैमेज टू द मेम्ब्रेन. सेल मेम्ब्रेन जो है उसको डैमेज करेंगे तो क्या होता है, सेल का अंदर का कंटेंट जो है वो बाहर आ जाता है, मतलब बैक्टीरिया मर जाएगा. सो दे डैमेजेस द cell wall. So they damage the cell wall and releases the cell content. Next one is ethylene oxide. So ethylene oxide का mechanism क्या है, mechanism देखो यहाँ पे. So it causes the alkalization of amino acids, carboxyl, hydroxyl, sulfhydryl groups of protein molecule. तो ये क्या करता है, alkalization करता है. So it will alkalize the carboxyl end, hydroxyl end, sulfhydryl end of protein molecule. Halogens, this is chlorine, iodine, and bromine. So what they do? Iodine is a strong oxidizing agent. Chlorine is also strong oxidizing agent. Chlorine is used for disinfection of water. Chlorine is used for disinfection of water, and this iodine is used as a antiseptic and disinfectant on the human skin. Heavy metals. So heavy metals denaturate the enzymes and other essential protein. So they are also antiseptic. Surface active agent, anionic and cationic detergents. They causes the disruption of enzyme protein, denaturation, disruption of plasma. And आपके surfactants होते हैं, anionic, cationic, this is sodium level. And they are also antiseptic. They also kill the, है ना, आपने देखा होगा कोरोना वायरस के टाइम पे कि आपको it is not necessary to use alcohol based sanitizer. आप simply soap water भी use कर सकते हैं. Why soap water? Because anionic, cationic detergents are present in the soap. So they also disrupt the enzymes, proteins, denaturation, and disrupt the plasma of bacteria and virus. So they are used as a, you know. So yeah, the point. Next we'll see methods of sterilization and their respective indicators, biological indicators. Very very important. Methods of sterilization and their respective indicators. मतलब क्या कि स्टरलाइजेशन हुआ है या नहीं है. आपने क्या किया, आपने आपके ग्लास वेस वाटर ओवन में रिक्वायर्ड टेम्परेचर पे रख दिए, बट आपको कैसे पता कि ये पूरा स्टरलाइज हो चुका है नहीं. फॉर दैट यू नीड टू इवेल्युएट. सो मेथड्स ऑफ स्टरलाइजेशन एंड देयर वैलिडेशन, you need to validate that method. So by using indicators, So different different indicators are used, फिजिकल इंडिकेटर्स, केमिकल इंडिकेटर्स एंड बायोलॉजिकल इंडिकेटर। इसके ऊपर है बहुत बार क्वेश्चन आ चुका है, इंपॉर्टेंट है ये चीज। सबसे पहले क्या है, ड्राई हीट स्टरलाइजेशन। ड्राई हीट स्टरलाइजेशन, सो फिजिकल मेथड्स कौन से, फिजिकल इंडिकेटर कौन से, सो फिजिकल इंडिकेटर्स आर मतलब रिकॉर्डिंग ऑफ टेम्परेचर बाय फिजिकल मींस, दैट इज द टेम्परेचर चार्ट्स। देन केमिकल मेथड कौन सा है, केमिकल मेथड इज नोन एज ब्राउनिंग ट्यूब। इसके ऊपर क्वेश्चन आ चुका है, ब्राउनिंग ट्यूब, ये दो बार क्वेश्चन आ चुका है। Brownian tube is a chemical indicator for dry heat sterilization. Dry heat sterilization, green spot आता है इसमें, है ना। Sterilization, if sterilization is incomplete, green spot आता है। And Brownian heat sensitive tips are there. They are used for the chemical validation. And biological indicators are nothing but the spores of Bacillus subtilis or niger. इसके ऊपर 3 बार क्वेश्चन। For dry heat sterilization, what is the biological indicator used? So it is a spores of Bacillus subtilis or spores of Clostridium tetani. They are used as a biological indicator. तो फिर से एक बार बोलता हूँ, तो आपने जो स्टरलाइज किया है, ड्राई हीट यूज कर रहे हो आप, सो आपको कैसे पता कि वो स्टरलाइज हो चुका है नहीं? सो फॉर दैट इंडिकेटर्स आर यूज्ड। सो फिजिकल इंडिकेटर, केमिकल इंडिकेटर एंड बायोलॉजिकल इंडिकेटर, 3 टाइप के होते हैं। फिजिकल इंडिकेटर मतलब क्या, रिकॉर्डिंग ऑफ टेम्परेचर बाय फिजिकल मींस, टेम्परेचर चार्ट्स। केमिकल इंडिकेटर में आता है ब्राउनिंग्स ट्यूब, green spots they are observed. Browdic heat sensitive tips are there. Browdic heat sensitive tips, they are used as a chemical indicators. Third होता है biological indicators. So spores of Bacillus subtilis, spores of Bacillus subtilis और Clostridium tetani. डिटेल मेथड जो होता है वो बुक में दिया है, इफ यू वांट यू कैन गो एंड रीड हाउ टू वैलिडेट, है ना। आपको ड्राइट में पर्टिकुलर ड्राइट से स्टरलाइज करना है बाय सबसेलिस स्पोर्स, बाद में उसको इनोकुलेट करके ग्रोथ मीडियम में आपको उसको ग्रो करना है। अगर बैक्टीरिया ग्रो होता है तो वो स्टरलाइज नहीं हुआ है, नहीं होता है तो मतलब स्टरलाइज हुआ है। तो पूरा प्रोसेस बुक में दिया है। यहाँ पे आपको इंपॉर्टेंट क्या है कि फिजिकल इंडिकेटर कौन सा है, केमिकल क्या है, बायोलॉजिकल क्या है, राइट। Am I audible to you? Am I audible to you? Second is nothing but a moist installation, क्योंकि क्या है, फास्ट लेना पड़ेगा, नहीं तो फिर क्या है, सिलेबस कंप्लीट नहीं होगा। सो डेली अपना अभी चालू रहेगा लेक्चर डेली। सो moist installation. So what are the indicators for moist installation? So biological indicator is nothing but the spores of Bacillus stereothermophilus, then Clostridium sporogenes. These are the used as a biological indicator. And then physical indicator is nothing but the autoclave tapes, thermocouples, they are used as a physical indicator. Then again sterilization by radiation, उसमें indicators क्या रहे, chemical dosing metals, they are used for validating the chemical indicators say there is a chemical dosing metals. And for biological, Bacillus pulmis and Cephreus are used as a biological indicator. Spores of Bacillus pulmis. गैस इंस्टॉलेशन में इसके ऊपर क्वेश्चन आ चुका है। रोसी सैचेट केमिकल रेडिएशन मेथड, मतलब केमिकल इंडिकेटर है रोसी सैचेट एंड बायोलॉजिकल है बैसिलस सबटिलिस स्पोर्स। स्टरलाइजेशन बाय फिल्ट्रेशन, स्पिडोमोनास डिमेटोरा एंड सेराशिया नार्सेंस, they are used as a biological indicator. Membrane filters indicator, they are checked by membrane filter का physical indicator है। bubble point test होता है दूसरा, forward flow test. These are the some methods of sterilization and their physical, chemical and biological indicators respectively. Very very important part है, एक दो बार देखेंगे और then see over there. Sterilization technique, moist heat, इसके लिए biological indicator कौन सा है, biological indicator is nothing but Bacillus stereothermophilus or Clostridium perfringens. They are the spores of this bacteria, biological indicator. और chemical indicator कौन सा है, स्ट्राइप पेपर, हीट सेंसिटिव स्ट्रिप। ड्राई हीट के लिए क्या है बायोलॉजिकल इंडिकेटर, स्पोर्स ऑफ बैसिलस सबटिलिस। केमिकल इंडिकेटर क्या है, ब्राउनिंग ट्यूब और ब्राउनिडिक हीट सेंसिटिव टिप्स। गैसेस स्टरलाइजेशन और इथिलीन ऑक्साइड, इसके लिए बायोलॉजिकल इंडिकेटर कौन सा है, बैसिलस पुल्मिस स्पोर्स। इसके लिए केमिकल इंडिकेटर कौन सा है, रोसी सैचेट। रेडिएशन बाय रेडिएशन और फिल्ट्रेशन स्टरलाइजेशन, इसके लिए देखो Biologically integrated is nothing but the Pseudomonas demetriae spores or Serratia marcescens spores और Saccharomyces cerevisiae. तो इसके लिए रेडिएशन के लिए ये इसके ऊपर क्वेश्चन बन चुका है Saccharomyces cerevisiae. Chemically integrated is dosimeters. Types of brownies tube. Brownies tube की भी types होते हैं. Brownies tube. So type 1 is used for the moist sterilization. Temperature होता है 126 degrees Celsius. You will get a black spot after complete sterilization.